Okay, let's dive in. Today we're looking at source material covering, well, a really huge development in molecular medicine. We're talking about a breakthrough in cancer treatment, a new drug. And it doesn't just, treat the disease. It actually targets and destroys the RNA that some aggressive cancers use for their, quote, immortality.
It really is a massive shift. It doesn't happen often, right? For so long, the main focus in drug development has been proteins. But this work, and we're pulling from research just published in Advanced Science, it shows a completely new way, a path to fundamentally tackle the disease at its genetic roots, moving beyond those, let's say, traditional targets.
Right, and our mission today is to unpack that research. It's led by Dr. Raphael Benamou and his team over the Hebrew University of Jerusalem. We want to make it clear why this is such a potential game changer. The core idea is they've built this small molecule. It acts like a sort of guided missile, designed to eliminate one specific RNA molecule that cancer hijacks to grow endlessly.
Exactly. Being able to zero in on a tiny, fleeting, but incredibly powerful RNA molecule. That's the definition of precision medicine right there. We're essentially watching scientists figure out how to dismantle the disease's molecular command center.
Okay, let's start there, the command center itself. If we're going to understand the fix, we need to understand the target. This breakthrough targets something called TERRA. So why TERRA? Why did it get this nickname, Immortality RNA?
Well, TERRA is fascinating. Normally, its job is actually protective. It's an RNA molecule, and it plays a role in keeping up telomeres. Telomeres, are the protective caps on the ends of our chromosomes. They safeguard our DNA, keep cells stable, super important.
And those caps, the telomeres, they naturally get shorter every time a cell divides.
It's normal.
Right.
Yeah.
Until they get too short, and the cell gets the signal to stop dividing, basically, to age and die off, the healthy process.
Precisely. And TERRA is part of the machinery that manages that whole process. But in certain really aggressive cancers, we're talking brain cancers, bone cancers, the really tough ones to treat the cancer cells, figure out how to hijack this system. They exploit TERRA to keep rebuilding their telomeres over and over. That lets them just ignore the natural aging and death signals.
Okay. So TERRA itself isn't inherently bad. It's just that cancer finds a way to sort of weaponize it. The cancer cells basically find the chemical override switch for cell division, giving them that limitless growth.
That's a great way to put it. In these aggressive cancers, TERRA becomes the key structure supporting the cancer cell's ability to just keep going indefinitely. So the thinking is, if you can stop that telomere maintenance, you could potentially force the cancer cell back onto that normal aging pathway, doom it to die, essentially.
Okay, but here's the tricky part, right? And this is where the science gets really amazing. RNA molecules. They're small, they don't stick around long, and lots of them look pretty similar structurally. How on earth do you design a drug that only destroys the rogue TERRA that's helping the cancer, but leaves the normal TERRA in healthy cells alone? That sounds incredibly difficult.
That difficulty is exactly why RNA has been called undruggable for so long. Traditional small molecule drugs, they usually need a nice stable pocket on a protein to bind to. RNA is different. It's floppy, constantly changing shape. So yeah, it needed a totally new approach. And that approach is this technology called RIBOTAC.
RIBOTAC. Okay, that stands for Ribonuclease-Targeting Chimera. Chimera.
Yeah.
That suggests it's like built from different parts. What's this chimera molecule supposed to do?
Think of it like a tool with two distinct ends. One end is for recognition, finding the target. The other end is the, let's call it the destruction module. The researchers knew they couldn't just use a general chemical hammer. They needed extreme specificity, hyperspecificity, really.
Okay, so the recognition end. What exactly is it looking for? How does it find the right TERRA?
It's designed to recognize a very specific, accessible shape that the malfunctioning TERRA molecule folds into. It's a structure known as a G-quadruplex.
G-quadruplex. Okay, that sounds technical. Can we break that down a bit? What makes that shape so special that this drug can find it and ignore everything else?
Right, so G-quadruplexes, they're basically four-stranded structures made of nucleic acids, different from the usual double helix we think of with DNA. Now, these shapes can pop up elsewhere too, but the key here is the engineering. The drug was designed to recognize the specific geometry of the G-quadruplex within the TERRA molecule. So it's not just any G-quadruplex. It's about how it's presented, its context within TERRA. It creates this unique lock-and-key situation. That makes it distinguishable from other RNAs, even if they have similar bits. That's the real genius here.
Got it. So the RIBOTAC molecule finds that specific G-quadruplex shape on the rogue TERRA, locks on. Okay, check. Then what? What does the other end, the targeting part, do?
Exactly. Once it's latched on, it essentially sends out a signal. It recruits something that's already inside the cell to do the actual demolition work.
And that something is.
It's a natural enzyme the cell already has called RNase L. This is really crucial. RNase L is actually part of the cell's own innate immune response. It gets activated when the cell senses viruses or major stress, and its job is to chop up RNA. The RIBOTAC molecule basically acts like a molecular beacon or maybe a magnet, pulling RNase L right next to the TERRA molecule it's bound to.
Wow. Okay, so instead of putting in some toxic chemical to kill the RNA, they've engineered a molecule that basically hijacks the cell's own defense system and points it like a weapon at a specific internal threat, the rogue TERRA. That's incredibly elegant, solves that whole precision problem.
It really is. It's leveraging the cell's biology instead of just fighting against it. And the research confirms this works with amazing precision. The compound was highly selective. It successfully targeted and degraded the TERRA RNA without causing much collateral damage to other important RNA molecules, even ones that might look structurally similar in places.
Okay, let's talk results then. Precision is fantastic, but did it actually work? Where did Dr. Benhamou, Elias Keskia, Deepak Dahatande, and the team test this RIBOTAC molecule?
So the initial tests, the proof of concept, were done in cancer cell lines in the lab. Specifically, they used HeLa cells and U2OS cells. And it's important to note, both of these cell lines come from really aggressive, hard-to-treat human cancers, so they're good models for the kind of challenging diseases this approach might eventually target.
Aggressive cancers, okay.
Right.
And what did they find in those cell cultures? What happened when they treated them?
The results were really encouraging, especially for this early stage. The treatment definitely reduced the levels of TERRA inside those cancer cells. And critically, that reduction directly led to a significant slowdown in the cancer cell's growth. So, well, yeah, it's still cell lines, not whole organisms yet. The mechanism worked. It showed proof of concept, destroy TERRA, slow down the cancer's immortality engine.
That link destroying the molecule leads to slower growth. That's the key finding, isn't it? But this brings us back to that paradigm shift we talked about at the start. Why is targeting this specific RNA so revolutionary? Most cancer drugs we hear about target proteins, right?
Yeah, it really comes down to the hierarchy in biology. You know the central dogma. DNA makes RNA, and RNA makes protein. Proteins are the workhorses, the enzymes doing jobs, the structural bits. Most drugs are designed to physically block a harmful protein after it's already been made.
Right. You're fighting the end product.
Exactly. But RNA, especially these long non-coding RNAs like TERRA, they often act more like the blueprint or the regulator or maybe the foreman at the construction site. By targeting TERRA, you're stepping in much earlier in that chain of command. You're going after the underlying mechanism, the thing that allows the cancer to maintain itself, rather than just mopping up the downstream effects of the proteins it causes.
I get it. So targeting the RNA like destroying the faulty instruction manual for the factory instead of trying to disable every single malfunctioning machine on the factory floor one by one.
That's a pretty good analogy, yeah. And if you think bigger picture, this potentially unlocks a whole range of therapeutic targets that were basically off limits before. Lots of genetic diseases, debilitating conditions involve regulatory RNAs that don't even code for proteins. They were considered undruggable. This RIBOTAC tech suggests maybe, just maybe, we can now develop treatments for diseases previously thought impossible to reach with standard drugs.
Okay, that sounds incredibly promising. But let's just pause for a critical look. The selectivity, the mechanism, it sounds brilliant. But what are the big hurdles now? What are the limitations? We're still talking cell lines here, right?
Absolutely we are. And that's the primary limitation, as always with preclinical stuff. Moving from the dish in vitro to a living system in vivo. That's the big jump. A cell line proves the mechanism works chemically, but it can't tell you how the drug will get processed by a whole body, how it gets delivered efficiently to the right tissues, or if it's tolerated long-term by a complex organism. So the next steps, undoubtedly, will involve really rigorous testing in animal models to figure out the pharmacokinetics, how the drug moves to the body, and the toxicology, is it safe?
That makes total sense. The jump from a petri dish to, say, a mouse, let alone a human, is huge. But the core finding still stands, doesn't it? This is proof that we can engineer these highly specific molecules to basically co-opt the body's own defenses against rogue RNA. It feels like a fundamental shift in what's possible therapeutically.
It really does feel that way. The discovery that this small chemical, this chimera, can specifically bind to that RNA G quadruplex in TERRA and activate RNase L that sets a powerful precedent. It's a template, one that could likely be adapted to go after other crucial support structures and other diseases at that fundamental molecular RNA level, far beyond just these specific cancer cells.
So for you listening right now, what you've hopefully grasped is this major shift happening in biology and medicine. Understanding the target TERRA, the cancer's so-called immortality RNA, and understanding this ingenious tool, RIBOTAC, used to selectively take it apart by essentially weaponizing the cell's own cleanup crew.
Yeah, the development of the specific small molecule, the one detailed in advanced science, it really feels like a signpost, suggesting we're entering an era of maybe ultra-precision medicine, where the focus shifts from just managing symptoms or blocking the final protein products towards actually eliminating the fundamental genetic instructions, the RNA instructions, that allow the disease to thrive in the first place.
So here's something to think about. If targeting the RNA that controls disease is the future, and we now have this method, RIBOTAC, to selectively activate the body's own RNA-destroying enzyme, RNase L. What previously untreatable genetic disease out there, maybe one driven by some obscure, undruggable regulatory RNA, might be the next big target for this kind of technology. Something to ponder until our next deep dive.